very much for the invitation to speak and for the kind introduction. The slides look okay?
Yeah, we can see that.
Great. All right. And please feel free to either unmute yourself and ask questions or type them in the chat while I'm talking. So I'm going to talk to you today about a paper that we just had published in Nature Communications, which has to do with mosaic integration of single-cell data. As you may be aware, there's been a revolution in recent years in the ability to measure different molecular features of cells at cellular resolution and within tissues. And so a key question in the field today is, how can we use all the information from these distinct measurements to define cell types in an unbiased fashion? And so there's an increasing need for computational approaches that can take distinct data sets, each of which may measure some different subset of the features shown on this slide. And how can we use all of these data types to move toward a unified definition of cell types in terms of their molecular features? So my research group has been working on this challenge for several years, and we've developed a set of tools called LIGER, which stands for Linked Integration of Genomic Experimental Relationships. And we have several publications in this area. So I'll just give you a quick overview of kind of the work that led up to this particular Nature Communications paper and then go into that. So LIGER actually started during my postdoc at the Broad Institute with Evan McCosco, when we were thinking about how to integrate single-cell RNA-seq data sets from different regions of the brain and from different species and also different human subjects. And we came up with this idea of using integrative non-negative matrix factorization as a way to identify shared and data set specific features that we can use to identify the similarities and differences of cell types across different experiments. And the heart of the LIGER framework is this matrix factorization, where we have a set of gene level features that are shared across data sets. And then we have a set of data sets, which could be gene expression measurements from different individuals or different species, or data sets of different omics types, such as gene expression, spatial transcriptomics, and epigenetic measurements. And integrative non-negative matrix factorization takes all of these data sets and jointly decomposes them into a set of data set specific and shared metagenes, which are then used to find a shared latent space in terms of K metagene expression levels, which can then be used for joint clustering to identify shared cell types across data sets. And this is kind of the heart of the approach that we build on in the Nature Communications paper. In a subsequent publication last year, just before the Nature Communications paper was submitted, we developed an extension to the LIGER framework that allows iterative single-cell multi-omic integration using online learning. And our basic innovation in this paper was to derive a new algorithm for integrative non-negative matrix factorization that allows us to process the data in mini batches. And this has several advantages. One is that if you have a really large data set, you don't have to store the entire data set in memory. You can just load it from disk, one mini batch at a time. And this allows much more efficient factorization in terms of both the time required and the memory usage. The second big advantage is if you have data that's actually coming in a streaming fashion, you can iteratively update the factorization without having to recompute from scratch every time a new data set arrives. And this iterative capability is also useful if you have data of questionable quality or that you're not sure how it relates to the existing data. You can use scenario 3 to project the new data into the factorization that you've obtained from scenario 1 or scenario 2 without changing the metagene factors. The challenge that we wanted to address in the Nature Communications paper is how can we integrate data sets that have partially overlapping features? And so we developed a new algorithm called unshared integrative non-negative matrix factorization, or UINMF, to address this challenge. And this was work that was done exclusively by my PhD student, April Kriebel. So a little bit more detail about the INMF framework, which is the foundation for this UINMF approach. If we have our cell by genes matrix, matrices that have N1 or N2 cells and G genes, we can write an optimization problem shown on the right here to formulate how to find the optimal cell and gene factors. And then we can solve this optimization problem using block coordinate descent. And at the end, we obtain the matrices HI, one for each data set, which give us cell factor loadings. The matrices VI, which are data set specific metagenes, again, one for each data set. And then a set of shared metagenes W that are identical across all data sets. And the inclusion of this shared metagene matrix is really kind of the secret sauce that allows the H's to correspond across data sets, because we have this regularization penalty that minimizes the amount of the reconstruction that comes from H and V, which has the effect of maximizing the amount of information that's in the shared metagenes. Another way of saying this is we find a set of cell factors that are constrained to have the same signal across data sets. And so this allows us to identify cell types and also biological and technical continuous factors that are shared across data sets. Another really nice advantage of this integrative non-negative matrix factorization framework is that the shared and data set specific metagene factors are often biologically interpretable. So we showed in our cell paper that, for example, if you look at this gene expression data from the human substantia nigra across multiple donors, we can identify a particular factor like this factor 28, where the metagenes, the shared component of the metagene factor contains signatures of, I believe it's in this case, activated oligodendrocytes. And if we look at this data set specific metagenes, they actually contain signatures related to the disease status of the patients. In this case, one person had some kind of post-mortem diagnosis of early neurodegeneration, and the other individual died of head trauma. And so there are some genes upregulated related to that traumatic event. And so the starting point for the Nature Communications paper was basically a key limitation of INMF, which is that it only uses shared features between data sets. And this is also a limitation for commonly used approaches like Seurat and Harmony. The first step in processing the data is to identify the shared features between data sets that you're integrating and to throw everything else away. This is a problem that was also recently identified in a review paper, a really nice review paper that appeared in Nature Biotechnology, where the authors identified 4 different types of integration, 4 different integration scenarios. The first scenario is they called horizontal integration, which is basically where you have the same features measured in multiple different data sets. So single-cell RNA-seq data sets all measure the same genes, for example, and so you can use those shared genes as the basis for integration. Liger, Seurat, Anchors, and Harmony all use this strategy. Another scenario is vertical integration, where what's shared across data sets is the cells themselves. And approaches like MOFA and Seurat, weighted nearest neighbors, solve this problem, where, for example, you may have proteins and gene expression measured in the same cell, and you want to use those protein and gene expression matrices jointly to do dimension reduction of the set of cells. A third scenario is called diagonal integration, where each data set measures a different type of feature, and there's neither shared cells nor shared genes. And there are several approaches for this, but in general, these methods are not as robust as the approaches that rely on shared cells or features. because they have to rely on some shared underlying geometry, which it can be a little bit more tricky. And then in this Nature Biotech review paper, the authors identified a fourth type of data integration, which is sort of a mixture of horizontal, vertical, and diagonal integration. And they called this Mosaic Integration. And the idea is basically that in Mosaic Integration, you have both shared features in some cases and shared genes, or sorry, shared cells in other cases. But there are a number of differences in the features that are measured across your data sets. So another way of talking about Mosaic Integration is basically to say that you have data sets with partially overlapping features, where some features are shared and some are not shared, and the features that are shared may differ across the different data sets. So this Mosaic Integration scenario is basically the motivation for our UINMF algorithm. And just to restate the problem in a different way, using only shared features and treating the data sets like we're doing horizontal integration has the disadvantage that it often requires discarding important data. So for example, if we're going to integrate gene expression and chromatin accessibility data from single cells using Liger or Seurat, we have to throw out intergenic features that are not linked to a particular gene from the attack data, even though these features can often be very important for distinguishing the chromatin accessibility landscape of different cell types. Another example where this is problematic is if we're trying to integrate a targeted spatial transcriptomic approach like MERFISH or StarMap that can only measure a subset of all genes because they use targeted capture probes. And so in this case, if we're going to use one of the existing methods, we would have to throw out all of the genes from the RNA-seq data that are not measured in the targeted spatial data. Another scenario could be if we're trying to integrate targeted spatial data with a multimodal measurement like 10x multi-omic that has both gene expression and chromatin accessibility in the same cells. And an especially interesting case is where we are trying to integrate across species, and we have both orthologous genes that are clearly mapping in a one-to-one relationship across species, but we also have non-orthologous genes which may have many-to-many mappings across species or no clear orthologs. So this slide is going to kind of serve as the outline for the rest of the talk, where I'll show how UINMF can help in each of these four scenarios. So the core of the UINMF method is basically changing the matrix factorization approach of INMF by incorporating unshared features that may occur only in one data set. And basically the trick we use to do this is to concatenate a U matrix to the V matrix. Recall that V is dataset specific metagenes. And then this U matrix corresponds to unshared metagenes. And to make the dimensions of the matrix factorization work out, we have to also concatenate a zero matrix with W so that this matrix product is then V stacked with U added to W stacked with 0 and then multiplied by H. And then we can update the optimization problem as shown below so that now we have stacked W with 0 and stacked V with U. And we also have corresponding data, which we call P, which are different size matrices potentially for each data set, and we have to stack those in the matrix factorization. And then we also update the regularization part of the optimization function here so that we're minimizing the amount of the reconstruction that comes from both the dataset specific metagenes and the unshared metagenes. And one way to think about this intuitively is that now we can factorize jointly data sets that have neither the same number of rows nor the same number of columns. So it's kind of a unique matrix factorization approach. And so the heart of the UINMF method is really this U matrix, which allows us to incorporate unshared features. And it's really flexible because we don't have to actually know anything about how these unshared features relate to the shared features. And this is nice because often, like for example, with intergenic peaks, we don't necessarily know which peak which gene each peak should link to. And so there are some approaches out there where if you assume a particular linkage, maybe from TF motif information or something, you can use that as the basis of the analysis. But here we don't have to assume that. And so the unshared features could be intergenic peaks or genes that are measured in one data set but not another, or non-homologous genes, for example. And then we iteratively update the U matrix in conjunction with V, W, and H using block coordinate descent. So just a few words about how the algorithm actually works. This is a form of non-negative matrix factorization. For simplicity, I'm just going to present how the algorithm works using only a W and an H matrix, but we also have V's and U's in our case. So basically, the typical strategy to solve an optimization problem like this is to use an iterative minimization strategy. Some people use multiplicative updates. We chose to use block coordinate descent, which has more firm theoretical grounding. And basically what that means is we solve first for W. We find the optimal W given a random starting point for H. Then we update W in that way and fix it, and then update H using the best W and iterate that process until we reach convergence. And this is a nice strategy because it's conceptually simple. The updates are relatively easy to derive analytically, and we have a theoretical convergence guarantee that if we're finding the optimal W given H and the optimal H given W at each step, we're guaranteed to converge to a local minimum at the end of the optimization. And then the last detail that is needed to make this whole thing work is actually a way of solving for W and H at each step. And we do that using non-negative least squares. So basically we use a numerical solver that solves for W holding X and H fixed. And that turns out to be a least squares problem with non-negativity constraints. And if anyone has questions, I'd be happy to talk in more detail about that. But otherwise, I will press on to the applications of the method. So the first application is integrating single-cell RNA-seq with single nucleus ATAC-seq data and using intergenic peak information. So we perform this analysis on a snare-seq data set, which is a data set that actually measures gene expression and chromatin accessibility in the same cells. And this is a case where, because you're measuring both things in the same cells, you don't strictly need to use an approach like UINMF. But we picked this data set because it gives us ground truth for assessing how much information the intergenic peaks can add to the analysis. So our strategy measurements that were performed on the same cells and split them and treat them as if they were measured on different cells, and then see if we could recover the held out relationship between the gene expression and chromatin accessibility. And in this case, we used UINMF with 3 matrices. For the gene expression data, we have a cells by genes matrix. We also have a cells by genes matrix for the ATAC-seq data, where we calculate a pseudo expression value using the chromatin accessibility of each gene. And then we also have a matrix of intergenic peaks, which are selected to be not overlapping the gene body of any of the genes in the red matrix. And so then we calculated 2 metrics, the alignment score and the FOS-CTTM score, which stands for fraction of samples closer than true match. And basically, our goal here was to figure out how the addition of intergenic peaks would change the ability of UINMF to identify corresponding cells between the 2 measurement types. So because these are the same cells, the theoretical maximum alignment score is 1, and the theoretical maximum FOS-CTTM score is 0. So we want a higher alignment score and a lower FOS CTTM. I guess I should explain what that metric is. Basically, because we know which RNA and attack profiles actually correspond because they were measured on the same cells, we can calculate the nearest neighbors in the factor space using the factors from UINMF. And we hope that cells that actually correspond in terms of their measurement are near each other in the factor space. So if a cell's nearest match is actually the corresponding profile that it was sampled from, then we've done well, and the FOS CTTM will be 0. If there are 2 cells that are closer than the true match, then we'll have an FOS CTTM of 2, and we can average those values across all cells in the experiment. So what we found is that if we add features, essentially the more peaks we add, the more the alignment score goes up, and the more the FOS CTTM score goes down. And we're showing these results with box plots because we did multiple random initializations, because the factorization can differ slightly depending on which random seed you use at the beginning. So this is encouraging because it means that incorporating the intergenic information improves the results. And we also did a comparison with our previous version of INMF and with Harmony and Seurat, and we found that we were able to consistently see better results compared to the INMF algorithm, which doesn't use the intergenic peaks. We got a better alignment score than Seurat and a slightly worse FOS CTTM score, but Harmony was clearly worse at its ability to align these data types, which makes sense because it was designed primarily for gene expression integration. We also just did a qualitative analysis to see if the cell types that we identified were corresponding to the labels that were published, and we found that they were quite well corresponding. And so that's the first application of UINMF. The second application is the one where we found the biggest benefit from using unshared features, and that is integrating spatial transcriptomic data and single-cell RNA-seq data. So spatial transcriptomic protocols are great because they give you spatial coordinates and gene expression. And so you can use information about which cells are physically next to each other in the tissue to learn about tissue function. But the problem is that a lot of spatial transcriptomic protocols lose something in terms of what they're telling you about gene expression. For example, the probe-based methods like MERFISH and StarMap and AwesomeFish and many others require designing probes to target your genes of interest. And many of these protocols can't target very many genes at once. So the challenge is that if we want to use an approach like Seurat or INMF to integrate data from this type of spatial transcriptomic platform, it requires throwing out most of the genes that are measured in the single-cell RNA-seq data. And particularly if you're looking at a protocol like AwesomeFish or this 3D version of StarMap that was published a few years ago, they only measure a few dozen genes, and so you have to throw away most of the transcriptome if you want to integrate them using only shared features. So we tested UINMF for this task where we took a StarMap data set for mouse frontal cortex that measured only 28 genes. And the reason the number of genes was so small is that it measured a three-dimensional block of tissue. And so they had some custom chemistry that allowed 3D measurement of genes, but it required a trade-off in terms of the number of genes detected. So we used UINMF to incorporate an additional 2,700 variable genes from the single-cell RNA-seq data set, and we put those in this P2 matrix, which we jointly factorized using UINMF. And then we did a head-to-head comparison to figure out how much benefit we got from incorporating those extra genes in the single-cell RNA-seq dataset. So what we're showing here are UMAP plots. The left one is UMAP of only NMF run on the StarMap data. The middle plot is UINMF using only the 28 genes that are shared between the StarMap and single-cell RNA-seq dataset. And then the UMAP on the right is the result of using UINMF with 2,700 variable genes plus the 28 shared genes. And you can see that clearly the cell types are much better separated in the plot on the right. I should say the colors are the published labels for the RNA-seq dataset, and the StarMap cells are shown in gray. If we look at the marker genes that each of these clusters expresses, we can confirm that we are correctly aligning the cell types between StarMap and single-cell RNA-seq. And you can see that, for example, the genes on this diagonal here have very high expression, and the expression is mostly exclusive to one cluster. So that confirms that the key markers for these cell types are matching between the measurement types. And if we do a systematic analysis of the adjusted RAND index and purity of the clusters compared to the RNA-seq clusters, we can see that there's a huge boost from incorporating those 2,700 genes compared to the approaches that use only the shared genes. So UINMF is shown in gray here, and INMF, Liger, Seurat, and Harmony are in blue, yellow, and red. If we look at the StarMap data, we can confirm that the spatial positions of the cell type assignments are also very reasonable and consistent with what we would expect. We can see the excitatory layers, excitatory neurons arranged in the cortical layers. We can see the distinction between medial ganglionic and caudal ganglionic eminence-derived interneurons, where the CGE interneurons tend to be in the upper layers. We can see that polydendrocytes and oligodendrocytes are also somewhat separated, and we can see differences in frequency between endothelial stalk and tip cells in different layers. And all of this accords well with what's known about the features of the cortex. So, oh yeah, and I should say, using the factors inferred from UINMF, we can also impute the expression of many genes that are not measured in the StarMap data, so that we can have approximate spatial expression profiles of these genes. And so here I'm showing two examples, including a marker of oligodendrocytes, which is primarily expressed in the white matter, and a marker of endothelial cells, which is expressed above and below the cortex. We also did a robustness analysis to show that if we vary the lambda parameter, which is the strength of the regularization penalty, or the K value, which is the number of factors, it doesn't strongly influence the performance as long as you stay within a certain range. We also explored what the unshared factors are learning in this case. And we found something interesting, which is that the U factors have their highest loading values on factors that correspond to non-neuron cell types. And we think this makes some sense because the proportion of neurons versus non-neurons is quite different between the two data types. And there are a lot more non-neurons in the StarMap data than there are in the gene expression data. We confirmed that UINMF provides a significant boost in a similar integration task where we took an awesome fish data set that measured 32 genes, again from mouse frontal cortex, and we used UINMF to include an additional 2,000 variable genes. So again, the unshared features are coming from the single-cell RNA-seq data set, And if we use only the shared features, we get a UMAP plot that looks like the one on the top. And if we incorporate the unshared features, we're much better able to resolve the different cell types that are present in the data set. Again, if we do a quantitative analysis of the purity and adjusted RAND index of the clusters that we get from the UINMF result compared to the published clusters from the single-cell RNA-seq data set, we find that there's a significant boost in both the purity and ARI compared to the approaches that use only the shared genes. And if we look at the labels for the awesome fish cells in their spatial context, we can again see that they have the expected biological features, including excitatory neuron layers, astrocytes in layer 1, which there's a special kind of astrocyte that we characterized in one of our previous papers. The white matter region down here is composed primarily of oligodendrocytes and polydendrocytes. And then in this particular tissue section, some of the subcortical regions were captured as well. And so we can see inhibitory neurons that indicate the presence of the caudoputamen and internal capsular regions. And we did a similar robustness analysis to show that the alignment score is quite robust to the K value and lambda value that you use. Okay, so I'm about to change gears and talk about another integration scenario, but first I wanted to throw in the supplementary analysis that one of the reviewers asked for, which turned out to be pretty informative. So one of the questions the reviewers asked us was, are there cases where incorporating unshared features makes the result worse or doesn't have as big of an effect? And specifically, can you trace the strength of contribution of the unshared features to the quality of the data set that they come from? And we found out basically that the answer is that yes, if you have higher quality unshared features, they have a bigger effect on the result. And to investigate this, we used 2 different protocols from the same tissue, a SMART-seq data set and a 10x version 3 3' capture data set from the mouse motor cortex. And if we had an expectation going into this analysis, it was that we thought the SMART-seq data would contribute more in terms of unshared features because there are more reads per cell, there's a better transcript detection sensitivity, and it's a full-length protocol as opposed to just 3' capture. And basically our analysis confirmed that that is the case, and that if we have unshared features from SMART-seq data, it significantly improves the result, more so than if we include unshared features from 10x data. So the specific analysis that we did was we reduced the number of shared genes from 500 to 100 in increments of 100. And then we incorporated unshared genes by selecting variable genes from either the 10x or the SMART-seq data set. And so if the unshared features came from the SMART-seq data set, we could see a pretty good preservation of the cluster purity compared to the clusters we get if we use all the variable genes from both data sets. And that's true even down to as few as 100 variable genes. Whereas for the 10x data set, we still see an increase compared to unshared features, compared to shared features only, which is in red, but there's a much steeper performance degradation as we get fewer and fewer shared genes. So basically this analysis indicates that the unshared features add more information if they come from the higher quality data set. And these are some UMAP plots showing the differences among 500 shared genes versus 100 shared genes, whether you're using no unshared genes or genes from SMART-seq or 10x. I see someone has their hand up. Do you have a question?
Yeah, I had a question on the unshared and the shared genes for the SMART-seq and 10x. So in that case, would you think that we should like exclude or like the 10x unshared genes during the integration approach?
So the results, let's see if I can go back to the previous slide. Basically, the results show that you never lose anything by incorporating the unshared genes, because the red box plot is always the lowest. But basically, it's just saying that you get more. Like if you have to choose one data type to bring in your unshared features from, you would want to choose SmartSeq, because adding the unshared features from SmartSeq gives you more information than adding the ones from 10x.
Okay.
Thank you. Okay. So the third application scenario I'll discuss here is when we want to integrate targeted spatial transcriptomic data with a multimodal data set like 10x multi-omic or SNARE-seq. And in this case, we've got two types of unshared features. We have intergenic peaks that are not present in the gene expression data, and we have unshared genes that are measured in the RNA-seq, but not in the targeted spatial transcriptomic. So in this case, we added these two matrices, the peaks and the unshared genes, to the UINMF integration. And this gave us a total of 4,000 unshared features, which included 2,600 unshared genes and 1,400 regions of intergenic highly variable chromatin. And we were able to confirm a good cell type correspondence with the originally published labels. And we found a significant boost in terms of the purity and adjusted RAND index compared to the version of the analysis that uses only the shared features. And if we look at the spatial context again of these star map cells, we can see that they show the expected spatial features of the tissue, including excitatory neuron layers, white matter, and endothelial and mural cells above and below the cortex. Okay, and we did a separate ablation study to see which added the most information, whether the unshared genes or unshared peaks or both gave the best result. And we found that each added something to the analysis so that the best result came from both unshared genes and peaks. All right, the last analysis scenario that I'll talk about is cross-species analysis. And this is a challenge because depending on how closely related multiple species are, there may be a number of genes that have either no orthologs whatsoever, or have a complicated many-to-many relationship due to like a gene family that's similar between species. And so existing approaches like Seurat or Harmony or Liger require you to use only the one-to-one orthologs when you're doing this analysis, but there can often be important genes within a species that define the difference among cell types, but do not have a clear one-to-one ortholog across species. For example, there's this data set from the Brain Initiative Cell Census Network, where they did a comparative analysis of motor cortex in human marmoset and mouse. And they found a number of differentially expressed genes that were species specific. So for our first demonstration of how UINMF can be useful for this, we took a nice published data set from the mouse cortex and the lizard pallidum. And mouse and lizard are sufficiently distant that they don't have exactly the same type of corresponding brain anatomy, but the pallidum is roughly similar to the mouse cortex, and so we should expect that there would be some similar cell types between those two samples. So for our UINMF analysis, we used 1,979 orthologous genes that have exact one-to-one orthologs. And then we also did variable gene selection in the lizard to identify 166 non-orthologous lizard genes that were important. And this is the UMAP that resulted from the UINMF analysis of the lizard and mouse. And you can see that the cell types are quite well integrated. And if we look at the published labels from each of the species, we can see that they correspond quite well the way we would expect them to. The astrocytes and ependymoglial cells are aligned. The excitatory and inhibitory neurons are aligned, and some of the non-neuronal cell types are well matched as well. And so if we compare this result to what we would get from using only the orthologs, we can see that we're less able to separate some of the important neuron types, particularly some of the excitatory neuron layers. And we can quantify this again using the purity and adjusted RAND index. And in this case, we have nicely curated cluster labels for both the mouse and the lizard. And so we could look at the purity and ARI of both species. And we found that UINMF was the best in terms of the overall retention of both the mouse and the lizard cell types. Harmony was an outlier here because it seemed to basically just retain only the mouse cell types at the expense of the distinctions among the lizard types. And that's likely because the mouse data set was larger of the two in terms of the number of cells. We also showed that you can do this with more than 2 species. And so to do that, we reanalyzed the human mouse marmoset motor cortex data that I mentioned just a moment ago. And we incorporated several non-orthologous genes, several hundred, from each species. So we used 2,700 orthologous genes as the shared features, and then we added several hundred non-orthologous genes as the unshared features. So in this case, each data set had a set of unshared features. And if we do this, we find that most of the cell types are quite well integrated, and we see very few cell types that are specific to one species. However, there's one clear exception, which is that some of the layer 5 IT excitatory neurons do not align. In particular, we found this cluster 9 that aligns between human and marmoset but has no mouse cells, and this cluster 13 that has only mouse cells. And when we dug a little deeper, we found that there's one particular cell type in the mouse that maps. Sorry, I guess I misspoke. There are actually some mouse cells in cluster 9, but there are no human or marmoset cells in cluster 13. And so it turns out that this mouse cluster that maps to the human and marmoset is one particular type of layer 5 extratelencephalic projecting neurons. And 3 other types of L5 ET neurons from the mouse do not seem to have homologs in the human and marmoset. And this is actually consistent with the results from the Nature paper. And we dug a little deeper and found that we were able to replicate the result from the Nature paper, where they showed that there were some key functional genes, including ion channels, that were differentially expressed between the conserved and non-conserved L5 ET species. So basically what we're showing here are violin plots of cluster 9 versus cluster 13 cells for some ion channels that are supposed to be shared, like KCNN2 and KCNA1, versus species specific like KCNH1. And so essentially this confirms that we're able to identify both these majority of shared cell types across species, as well as the cell types that show species divergence between mouse, human, and marmoset. And with that, I will conclude, and I'm happy to take any other questions. Thank you.
Thank you. So any question? If you have any question, raise your hand or just put your question in the Q&A. So give other people like a few minutes to think about the question. I have one question. When you do the tool comparisons, like say compare Harmony, your tool Surat, I was just wondering, did you also compare the running speed? Because for our data integration, we usually have 15, 20, single-cell RNA-seq data. It's actually time consuming to do the data integration also. So have you ever compared that also? Sorry, you are muted maybe.
Sorry about that. Yeah, we did include running time stats in the supplement. I can't remember if we benchmarked every method, but I would say Harmony will be the fastest, and ours is probably a similar efficiency to Surat, maybe slightly faster.
Okay, great, thank you. Did you have a question?
Yeah, so thank you, Dr. Welch, for the excellent talk. One of the advantages that I find for your method is the allowance for the unshared genes, right? And that helps us retain any important genes that are important for a rare cell type. But when you look at it, how does the amount of gene or the number of genes influence integration? For example, in your example, one of the slides you had targeted sequencing data and also the single-cell RNA-seq integration, right? So the number of features that comes from targeted would be lower and particularly small compared to the single cell. Like how does it affect the integration? And a linked question would be in an integration approach, what level of recomputation occurs, which you mentioned in your very early slides? Like, do you recompute every single step? Like, is just adding the data and then recomputing, or is it starting from somewhere in the middle?
Okay, so the first question, the number of genes and how does that affect the result? I would say it's a complex relationship. It's so certainly the data set that has more features, like the norm of the matrix product for that data set will be larger than the other data set. But that doesn't necessarily mean that the larger data set completely dominates the smaller, because both basically the objective function is the sum over all of the data sets. And so, for example, in the cross-species integration example here, with the lizard and mouse, the mouse data set was actually the larger. And so some of the tools like Harmony really amplified the effect of the larger data set. But we found that basically our approach was better able to balance the sizes, the relative contributions of the two data sets. I don't have a theoretical explanation for exactly why that difference occurs between ours and Harmony, but it may have to do with how they calculate their reduced dimensions. And your second question was like, how often do you have to recompute analyses? And I would say that really depends on what your application is. The case where we thought about it was in the context of a large consortium that's like continually generating new data sets. We're part of the BRAIN initiative. So we did find that we were having to recompute our results, fairly frequently. Whereas if you're a single lab investigating a single biological question, it may not be as important, and you may be able to just gather all of your data sets and then analyze them all at once and be done.
Okay, and you also showed that the using UINMF achieves better separation of the clusters. Like, how do you think the final annotations are better with UINMF, or do you think like certain number of cells now the annotations, like what fraction of cells would change annotation in your estimate?
Yeah, that's a little hard to assess, but we were actually more focused on getting annotations for the spatial data. And so the annotations for the spatial data were certainly improved by this analysis. And it's kind of a hard thing to validate because you don't have very many markers for each cell in the spatial transcriptomic data. But the thing that we found that was very encouraging was that we were better able to identify refined spatial populations, like populations with refined spatial locations, when we looked at our UINMF clusters compared to the original published analysis of the spatial data.
Okay, thank you.
Any other question? Nope. I think just feel free to contact Dr. Welch if you have any further question. Thank you, Dr. Welch, and very nice talk. And we're very glad to have you here. And have a nice day.
Thank you very much. Bye-bye.
Thank you. Bye.